I told you for transcription, as for replication, for transcription, RNA polymerase is required. And this enzyme is RNA polymerase. And this you can see DNA, and this is RNA releasing because of transcription RNA. And you can see this open loop here inside DNA, RNA polymerase. So inside RNA, you will see always there is certain parts of DNA will be opened. So you can see this open. The difference between DNA replication and transcription is that replication happens in both the strands. It happens during S phase and both strands, and the whole complete set of DNA will be duplicated. The end result will be complete duplication. Here a segment of DNA will be transcribed, and that segment, within that segment also only one strand will be transcribed. And this transcription events happens always, and in the cells, in permanent cell and in dividing cell during G1 phase. So you can see this basic differences. You can see here. I've already done this basic differences in table form. Now this RNA polymerases, bacterial RNA polymerase has 5 core enzymes. Core enzyme consists of 5 protein together. These are called beta 1, this is beta prime and beta, and this is called alpha 1 and alpha 2. Those both alpha are identical, and then omega. So beta will make the main component of enzyme that will cause enzymatic reaction. With this beta similar component in eukaryote, you can see this RNA polymerase and RNA polymerase 1. You can see RNA polymerase A1 and A2 here, B1 and B2. So you can see A1 for this and A2 for this, and for RNA polymerase 3, C1 and C2. So that is how you can see here. These are the polymerase set. This eukaryotic RNA polymerase are of 3 types for different RNA, for production of different RNAs, while prokaryotic RNA polymerase is only one for all RNAs. And you can see this RNA polymerase also have more subunits. This eukaryotic have more subunits in comparison to bacterial RNA polymerase. So this is the picture showing the structurally they look similar. Both RNA and DNA polymerase, bacterial RNA polymerase, eukaryotic RNA polymerase, they look similar. But the difference you will see is eukaryotic have more components in it, but the enzymatic part, main enzymatic part of both will be similar. Spencer beta prime and beta will form main enzymatic part. This is Spencer part, while in eukaryotic this 1 and 2 polymerase B1 for RNA polymerase 2, B1 B2 for polymerase 1, A1 A2 for polymerase 3, C1 C2 component will form this part. So these are the differences, basic differences between RNA polymerases between bacteria and in eukaryote. In eukaryote also RNA polymerase 1, 2 and 3, they are different, and purpose is also different. And you can see these are the mRNA. You have to remember this. I will ask question from this. Okay, I will ask question from this, which polymerase synthesizes which RNA. So I will ask question from this. So, and then I will also I may ask question from this. So differences, but this is more important. This is more important. Remember this. I will ask question from this. You can see this is RNA polymerase of bacterial RNA polymerase. The core enzyme is made up of 5, and sigma is, when sigma is there, this is holoenzyme. What is the function of sigma? Sigma help this enzyme bind with promoter region. So we will, and this polymerization happens in 5 to 3 direction utilizing 3' to 5' DNA template. We have seen that. So these are the product of all RNA. These are the product of DNA transcription. These are the process of transcription. Different RNA will be generated because of transcription only. And this RNA, synthesized RNA, can be classified into 2 regions. One is coding. This DNA, the coding region synthesizes mRNA. And then all RNA are synthesized by non-coding region. Coding means protein. that actually have a message for protein, and that is only present in messenger RNA. Other are non-protein coding. They are synthesized from non-protein coding region. That means non-coding region. All other RNA, tRNA, rRNA, snRNA, these are synthesized from non-coding region. You can see these three processes are there. Initiation starts with the binding of this sigma subunits to the promoter region, and then after initiation, sigma will be shut down, and then this RNA polymerase will polymerize RNA, and after polymerization, there will be termination. This polymerization is called elongation, and after this there will be termination, and after termination, this polymerized one will. This RNA polymerase will be removed from the DNA. So these three steps are steps. If you remember RNA transcription. So transcription you remember this way. And what is promoter region? Promoter, I have already told you. Promoter region is made up of about 40 bases long. And this is upstream. What is upstream and what is downstream? I have already explained. The upstream is the downstream nucleotide or the nucleotide from where protein is coded, from where protein is from where RNA is synthesized. mRNA ultimately will form protein. So the nucleotide from which RNA will be coded or RNA will be produced, transcribed, and then the nucleotide which does not code to which does not produce RNA, and it is above this. So these are called upstream and these are called downstream. Upstream are given minus numbering, and that is why minus 10 region, minus 25 region. This base consensus means the base are very similar in among the species. This bases sequence are fixed, and this is called consensus base sequence. First is at 10 region is a Prinbo, and this is called 35 region is called this is GC rich region. So this will not be melted. When promoter binds this part will be melted, and then after binding of this sigma to this promoter, and RNA polymerase will be recruited, and after recruiting, so there will be three events for like RNA transcription. First event is close complex. First the promoter will bind to this, the sigma factor will bind to the promoter region, then RNA polymerase will be recruited. This is first event. Now after binding, what will happen? This region is weak, so that is why this region will be melted. This is called open complex. Okay, this is second event. And so what is there in open? And this open complex will progress as this RNA polymerase progress. This open complex will progress with RNA polymerase. So I have described what is open complex. So open complex is this complex. It has got two strands. So only one strand this RNA will be synthesized. Transcription will be done on only one strand, and this strand is template strand. And template strand is also called non-coding strand antisense. Why it is called non-coding antisense strand? I already explained, because it is not similar to this sequence. But if you can see this non-template strands, the sequence is very similar to RNA sequence. Here sequence of this nucleotide is very similar to RNA. Nucleotide based sequence, if you can see, it is very similar to RNA. That is why this template non-template strand is called non-coding and sense strand, because it makes sense with RNA. That is why sense strand and coding strand. So I told you what is coding, non-coding, template, non-template. All I have explained this strand. So this is these two strands will be present in this transcription bubble, and this is called transcription bubble. So open complex, second events will be open bubble in the promoter region. This open this region will accompanied by this scrunching, will pull the DNA and then will escape this DNA promoter region. RNA polymerase will escape this promoter region, and pulling of this DNA utilizes ATP, and there will be small, very 2 to 10 nucleotide RNA synthesis, and this is called abortive synthesis. So, and then after this RNA polymerase escape this promoter region, now what starts is RNA elongation phase. You can see here. So this elongation phase whole elongation phase, this replication bubble will go with RNA polymerase. So RNA polymerase has 3 channel. I have already explained 5 channel. One channel you can see between 2 beta spincer, there is DNA, and then this one channel is for non-template strand, and this channel is for template. Template strand means the strand where RNA synthesis will happen. So this is template strand, and then from this channel RNA will exit. And actually this picture is showing the binding of RNA polymerase to promoter region through sigma unit. So and actually the same events will actually go along this. So this replication bubble will travel with this, sigma will be released, and then transcription will happen. You can see this. And now one thing is not shown here, NTP. So 5 channels are there, remember that. Now elongation phase. So we have, in last class we also did elongation phase. So in elongation phase I told you, during elongation this is RNA, this is DNA. So in DNA there will be loop. That loop will travel. That loop here, you can see this one, I will make one loop here. So this loop will travel with RNA polymerase. So RNA polymerase, what does RNA polymerase do? As RNA polymerase moves, so RNA polymerase unwind this 2 strand, and then at the same time it will synthesize first RNA, and then RNA will be released. Only few nucleotide will be attached to this DNA, and other are removed from the hydrogen bonding. So you can see every time in this complex there will be only 8 to 9 nucleotide bound to that, and then other nucleotide will be peeled off. Other RNA will be peeled out from the DNA, and then after peeling, this DNA will be made double stranded by RNA polymerase itself. So the same way it will progress. It will synthesize, and again it will progress, synthesize, progress, and at the final there will be production of one mRNA. So this is how it moves, and it polymerizes. This polymerization rate is about 15 nucleotide per second. 15 nucleotide per second. Okay. Then topoisomerase 2 handles when there is supercoiling. And open, I told you, open complex moves. First closed complex will change into, if during initiation only, it will become open complex, and open complex will move. And this is a very impressive enzyme. Why it is impressive enzyme? Because it unwinds the DNA in front. This unwinds the DNA as it moves here. In front it unwinds the DNA, and then re-anneal this DNA, rejoin this DNA through hydrogen bond, back DNA. This backward DNA, it will re-anneal, and it will progress, re-anneal, and again open, re-anneal, open, re-anneal, and this 2 function is done together by this RNA polymerase. So this is re-annealing of back nucleotide here, and the opening of nucleotide here will be done by RNA polymerase. So it dissociates growing RNA also. As I told you, peel off the RNA polymerase, and there will be always formation of only 8 to 10 nucleotide will be there in hydrogen bond. So it performs proofreading function also. So proofreading function is not as precise as DNA polymerase. Not as precise as DNA polymerase. Okay, because DNA polymerase has to copy the DNA very exactly. That is why DNA polymerase has, the proofreading function of DNA polymerase is very exact. In some text you will find that RNA polymerase, they write that RNA polymerase does not have proofreading function. What is proofreading, by the way?
It's an error
yes.
process.
Yeah, error correcting process. Once when wrong nucleotide is added, that nucleotide will be removed. Some textbook has this. Some textbook you will find that somewhere the RNA polymerase does not do editing. But I have I have the RNA polymerase does editing. This reference, I will give you reference from a book called Watson, the Watson Crick, the scientist who got Nobel Prize. The same scientist has written a book, Molecular Basis of Gene. So from this book, I have written this. Proofreading function is there in RNA polymerase. That is why you can believe this. Elongation, I told you elongation happens like this, and during elongation, if there is some wrong insertion, so insertion will be removed by this two type of editing. One is hydrolytic editing, and other is pyrophosphorylatic. So what is water is used to break the bond, then it is called hydrolytic editing. When pyrophosphate, this pyrophosphate is utilized, also utilized to break the bond, and this is called pyrophosphorylatic editing. So two types of editing can be done by this RNA polymerase: pyrophosphorylatic editing and then hydrolytic editing. So editing will be done, base removal will be done, and then reverse one base removal, then there will be addition of fresh base, and then this progress. So you can see there is RNA channel, exit channel for RNA, and it will cause repealing. You can easily conceive the concept. So there are channel for everything. So if it moves, there is some protein that will cause annealing, and there is channel for DNA, exit and entry. So at the same time, when it moves forward, there will be opening, like finger will do opening here, and then automatically there will be resealing, like that of zip. You can see zip, there will be opening and resealing. The same way you can see zip is there, the same way you can see opening will be done inside the RNA, this polymerase, and then there will be zipping backward and then opening forward, zipping backward, opening forward. So you can conceptualize this with zipping. Now let's talk about termination. What is termination? So termination happens through, I have told you initiation. How does 3 phases of initiation? Then elongation. What happens in elongation as it progresses? Only 10 nucleotides, about 10 nucleotides are only in base pairing. So there will be in aligning back DNA and opening of front DNA, and then movement of that replication bubble will be there, and this peeling of the RNA will be there. Now there is termination when DNA open reading frame. What is open reading frame? I have already explained. Open reading frame is the unit of DNA where transcription happens, where transcription starts and ends. This is called open reading frame. So when there is a stoppage in the DNA, then their termination will happen. And termination, there are two processes by which termination happens. One is row dependent process, other is row independent process. So this is called row independent. Sorry, there is independent. This is row independent process, other is row dependent process. What is row dependent? Row is a protein. Row factor is a protein. The protein is hexameric protein. And that hexameric protein. This hexameric protein is a donut type of protein. You can see the protein is like donut. Okay. So this protein can be loaded on the RNA. I will tell you how. This protein can be loaded on that. This is hexameric protein. And this protein is very similar to helicase enzyme. It is also helicase that you have studied in DNA helicase. It is very similar to helicase. Its function is helicase. Its also function is to break this hydrogen bond. So this row factor is nothing but helicase enzyme, RNA helicase enzyme, RNA DNA helicase enzyme. And it is an hexameric. The structure is also very similar to the helicase that we have studied in DNA helicase. Now this is row dependent. Row independent process, you will see there are specific sequence along the DNA. Along the DNA there are a specific sequence, and that two sequence are very opposite to this sequence. This sequence, number 1 sequence, is very opposite to number 2 sequence, so that this 2 sequence will form hydrogen bond with each other, and that is how it will form a hairpin. This RNA produced will form a hairpin loop. Let's see how does it happen. You can see this. So this is row independent. You can see these two sequences are there. Can you see this two sequence here? Sequence 1 and sequence 2.
Yes, sir.
Okay, so this is coding strand. Coding strand, the RNA will be very similar to coding strand. Now you can see the RNA you can read from this coding strand. So you can see this A. A is very similar to T. A and T. You can see A and T. In place of T in RNA there will be U. A and U will form double bond. G here opposite sequence C. G and C here transcribed RNA will form triple bond. Here C and G. This C and G will form triple bond here. And then CCC, GGG is there. So all will form triple double hydrogen bond. And then at last C is there. Then C with G. So you can see this two will loop like this. If it loops, it exactly fit each other and make hydrogen bond like this. And this is called hairpin loop. This is called hairpin. You must have seen hairpin. Haven't you? Hairpin also looks like this. So hairpin, if you see, hairpin is like this. So this loop is called hairpin loop. And this hairpin loop is formed. And after formation of hairpin loop, after this, you can see there are like this, there are 80 rich region. And when RNA synthesis happens on this A and U region, you can see this A and U will be able to form only double bond. And this double bond, continuous double bond will not be that stable. And this double bond, this hydrogen bond between A and U are weaker. And because of this weakness and formation of loop, there will be termination of transcription. And this transcription, for this transcription, there is no requirement of other protein. And this is called, and this actually, the termination is not done by protein. And this termination process is called row independent termination. You can see here. So this loop will be formed, and the loop will bond with certain protein, but this protein will secure this loop. It does, and then because of this weak bonding and loop formation, this RNA will detach from RNA polymerase. As soon as RNA detach from, go away from this RNA polymerase, then RNA polymerase also get detached, because RNA polymerase binds to DNA with this junction, RNA DNA junction. If RNA DNA junction is not there, this RNA polymerase will also lose the contact. So see, there is only one events. This row independent termination occurs because of formation of hair loop and because of this weak bond between A and U. Because weak bond cannot sustain. And hairpin loop, if hairpin loop formed inside the RNA polymerase, what will happen? This hairpin loop weakens this bond still more. And then because of this whole RNA polymerase, RNA will be detached from this DNA. Okay? Did you understand?
Yes, I did. Thank you.
Okay. So now this is RNA. This is row independent. Row dependent is very easy. So when this is transcribed, RNA is transcribed, there is a very specific sequence. This is called RUT sequence. RUT sequence. Okay. What is the full form of RUT? Row utilization site. So there is a sequence of some nucleotide where this row factor will bind. So row factor will bind to this, and row factor, what does row factor do? Row factor pull this RNA. Okay, pull this RNA, because row factor is helicase, and helicase utilize ATP to pull this RNA. And because of pulling effect of RNA, what happens? This RNA synthesis termination will happen. Simple. This will pull this RNA. Protein will pull the RNA. Hairpin and this weak bond will dissociate this RNA from this RNA polymerase and this template. Here, because of row will pull the RNA. Did you understand the difference between row dependent and row independent termination?
Yes, sir.
Okay. So remember these two types of termination, row dependent and row independent. So how many, this is the end of the RNA transcription process in bacteria. So how many processes are there for RNA transcription? Tell me. Let's summarize.
Three
steps. So what are these steps? First step is
initiation.
What happens in initiation? How many stages are there in initiation? Steps?
I think 3. The close complex.
First is binding.
Sorry, binding.
Binding to promoter through sigma. Then second is close complex. And then third is open complex. Okay, open complex. And promoter escape, abortive synthesis, everything happens here. Now what is second step?
Elongation.
What happens in elongation?
The elongating enzyme unwinds the DNA.
Yeah, unwinds and reseal the DNA, and then polymerize the RNA. Polymerize and then does editing. Proofreading at the same time. So 3 events. Now third function is, third stage is termination. What are the process of termination?
RH independent termination and dependent termination.
Yeah, row dependent and row independent. Row independent. What is row independent? Formation of hairpin loop. And row dependent is by protein that pulls the helicase, RNA helicase protein that pulls the RNA. So this is the summary of your transcription. Actually, transcription is nothing but gene expression. You can see gene, our gene and bacterial gene or any gene or human gene. Human gene express through transcription. So transcription is, in other words, what is the meaning of transcription? Transcription means gene expression. Okay, did you understand this? Transcription means gene expression. How does gene present itself? Through RNA, mRNA. mRNA if synthesized will synthesize protein. So transcription means what?
Gene expression.
Will you memorize this? This is very important concept. You will remember. Transcription means gene expression. So this is one question. Which of the following process is process of gene expression? Process is process of gene expression. So option is replication, transcription, translation, and there can be fourth option is post-transcriptional modification, post-translational modification. What is the answer? Transcription. Okay. So transcription is a process of transcription is the process through which gene is expressed. So let's see the difference between this eukaryotic and prokaryotic transcription. So you know Let's start from the RNA polymerase. You already know, I have already given the difference between RNA polymerase, eukaryotic and prokaryotic RNA polymerase. You know this. So what is the difference between this open reading frame? So open reading frame in DNA and polymerase and RNA polymerase. First, talk about this open reading frame also. So in bacteria, this open reading frame has polycystone. Polycystone means one cystone, one protein. One cystone, one protein. Okay, one cystone means cystone is equal to gene. So polycystonic, this bacterial has polycystonic. Once transcription happens for one mRNA, there will be that mRNA will carry codon for many proteins. This is called polycystonic. But this mRNA of eukaryotes will be monocystonic. Very important difference. Monocystonic. And that monocystonic will also contain introns. Because of this, it contain introns. You know what is introns? The distance, the island. These are exons are island in the mRNA, which code for gene. And this is the part that is between these exons are introns, and that does not code for protein. So this will be removed through splicing, and this all part 1, 2, 3, and 4. All this exon will be taken together. 4 exon will be put together. This becomes mature mRNA. So removal of this intron will happen through splicing. And this introns is present in eukaryote only, not in prokaryote. So monocystonic with introns. This is polycystonic without introns. So this and the promoter region, this 3 basic differences about the structure. First, structure of RNA polymerase, then the cystone, the open reading frame. Then in open reading frame, what is promoter region? So promoter region is more complicated here in eukaryote. And this prokaryote promoter region is very simple. We have done this promoter region. So you can see in promoter region, there are 250 bases are there. In addition to promoter region will be assisted by some other activator region and inhibitor region. So this region is more complicated in eukaryote. This is very simple in prokaryote. So promoter region is bit complicated in eukaryote and non-complicated in prokaryote. So initiation. So initiation, here you will see what is there in initiation. One transcription factor is there in initiation. What is that? Sigma factor. Sigma is there in initiation. Now here you will see six different type of transcription factors are there. These are called general transcription factor. Here 1, here is 6. Again more complicated. During elongation, more protein, other proteins are also other protein helps RNA polymerase during elongation process. But elongation is done by this RNA polymerase, one RNA polymerase present, and that RNA polymerase alone will do elongation. It does not require help in prokaryote. Simple, very simple. Here more protein will come to help this RNA polymerase during elongation also. Termination will be done by either by raw protein and some organism, some bacteria. The termination will be row independent. You know this. And here the termination will be done by this 2 protein. We will see this 2 protein about this protein. And then RNA mRNA splicing is present in eukaryote. It is absent in prokaryote. This post-transcriptional modification, post-transcriptional processing happens in eukaryote more, and it is absent in prokaryote. At least for mRNA, it is absent. Now, transcriptional regulation. So the regulation is very complex. So what is transcriptional regulation? Transcriptional regulation is making the transcription on, doing the transcription on, and transcription can be made off. So this switched on, transcription is switched on and switched off. This process is called transcriptional regulation. So transcriptional regulation is done by a complex mechanism of activator and repressors are there, and there will be histone modification. Here operand, simple operand are there. Operator region, the operator region is some DNA. This is promoter region. Operator region will be some segment. It will be segment here after promoter, and this protein will bind here, and then after some protein binds here, the RNA transcription will not happen, will be blocked. This is very simple. Gene regulation is very simple. Here gene regulation, that is transcriptional regulation, is a bit more complicated in our body. We require more protein, more DNA segments are there. So did you understand the difference between these two? In one word, if I have to say, the difference is complicated. The RNA synthesis process is a bit more complicated in eukaryotes. Regulation is also complicated than prokaryotes. Prokaryotic RNA synthesis is simple. It is a bit complicated. More proteins are there, more steps are there. And that are the differences. Did you understand this? Differences? So I will talk about this eukaryote only. I will go fast for eukaryotic transcription. You can see this. So I told you this bacterial. First, this bacterial is polycystonic. This is mRNA. You can see the same mRNA has different type of this coding region. So this green will code for one protein, this red will code for other protein, and this same mRNA has 3 genes, and this is called polycystonic mRNA. This is present in bacteria. So remember, this is not present in eukaryote. Eukaryote has only one cistron, but there will be introns, intron, exon. Splicing will be required. So this is the main difference. You can see promoter has about 250 nucleotides, more than 100 nucleotides. You can see this nucleotide difference. Promoter region is, you can compare this promoter region with bacteria. So bacteria has -10 and -10 here, and -35 here, -25 and -70. Upstream promoter will be present on upstream, and this is this. Now this consensus, this is also consensus sequence from our body to other eukaryote, simplest eukaryote, unicellular eukaryote. They will have similar region here. I told you the significance of this region. This is called this is also called Tata box, or other name is given for this eukaryote. This is called Goldberg-Haugnes. This is called Haugnes box. Haugnes box. There is Prinbo box for bacteria. This Haugnes box. Now you see cytosine rich and AT rich, cytosine rich. That is why it is not that feasible for melting. This is more feasible for melting. So this promoter, you can see a bit similar in, actually principally they are similar, but they are more larger in eukaryotes. And with this promoter, there will be other segment as well that will be helping this promoter. But in bacteria, only promoter is there. Okay, and this promoter will be controlled by somewhere. The gene will be here, and that is called operator region. And this is operon, and this is how it will be regulated. But here, a number of helping DNA segments will be there. You can see many helping DNA segments are there. This is promoter region. This is promoter region. You can see this is promoter region, and this promoter region actually include Tata box. And this promoter region will be held by other enhancer. You can see enhancer region and repressor region. There are many DNA segments. So we will not go about all the DNA. This can be very close to this promoter region, or it can be very distant from this promoter, because eukaryotic DNA is very large. So it can be closer to promoter region, or it can be very farther from this promoter region. So distant promoter region are called this distant regulatory elements are the further regulatory elements, and the closer regulatory elements are called promoter proximal elements. So there are these elements that will regulate this promoter region. So there are many a bit complicated. So you have seen this complication. So now this promoter, at this promoter region, this will help this promoter region. This will help proteins bind this promoter region. I'm not going into detail. Just remember that promoter region is more complicated. There are many more helping hands to recognize this. So this promoter region will be bound by certain proteins. That is very equivalent to sigma factor. These proteins are called general transcription factor. And the protein that binds here, they are a specific transcription factor. Transcription factors. So remember, transcription factors bind DNA and stimulates transcription. And those are called transcription factors. This is general transcription factor. So general transcription, these are the general transcription factor. Anywhere this is general transcription factor is equivalent to sigma factor. Remember, this is nothing but sigma factor in eukaryote. So there are 6. So 6 transcription factor will bind like this. This is transcription factor D. This has got 2 components. This is called Tata binding protein, and it has got transcription activation factor. So 2 protein will form 1 transcription factor. This is called transcription factor 2D. I am talking about mRNA transcription. Okay, this is called transcription factor 2D. Now with this, other A and B will come. So you can, I'll give you mnemonics. Bad will come together. Bad, friend, F-R-I-E-N-D, friend. E is hiccup. So your bad friend, if you are having E hiccup, your bad friend will come and irritate you to is hiccup. So anyway, funny mnemonics, but it will help you. So BAD, with this D, B will, A and B will come. With this D, A and B will come. So bad, and then friend, F, transcription factor F will combine with this RNA polymerase. And this RNA polymerase 2 is helped by this transcription factor 2F will help this RNA polymerase bind this bad component. So bad, friend, and this E, transcription factor, now E will come here. E will bind this upstream elements, and then H will come here, and H will help in melting the DNA and progress, and it will help in elongation, start of the elongation phase. So there are 6 transcription factor, and that is associated with RNA polymerase 2. These are called general transcription factor. These 6 are equivalent to sigma factor. Remember this. And these 6 are bad. TF, transcription factor D will attach first. D, A and B, bad. And then F will help attachment of this polymerization, RNA polymerase 2. And then E, transcription factor E will attach, and then it will recruit H, and then transcription will start. Did you understand this? Very similar to bacteria, a bit more protein, that's it. And that protein also attached in.
Can I know the difference between the complexes?
You knew the difference. You knew the difference. Okay, that's the more important thing. So that's the more important thing. The difference between pre-initiation bacteria and eukaryote. So you can go visit this PowerPoint and you can memorize those things if you want. So now you can see the elongation phase. This is elongation phase. And you know what is there in eukaryotic DNA? In prokaryotic DNA, there is no nucleosome. Okay. Do you see nucleosome in prokaryotic DNA? This nucleosome? No. There is a nucleosome in eukaryote. That is why don't you see this nucleosome are very difficult to go through during transcription. And that is why during transcription, many protein come to help this transcription to actually remodel this nucleosome and help this RNA polymerase to progress through this hilly areas. So actually DNA is very difficult. Many hills are here, many blocks are there. So the other proteins come to help this. Some protein will remodel this. This will actually unfold this DNA from this nucleosome. These are called chromatin remodeling protein. So this protein will take out this chromosome, and that is how this protein will help this RNA polymerase. Sometimes you see there are many mountains are there. So what will happen? Sometimes RNA polymerase will get tired. So there are these proteins, some proteins that are there to help this RNA polymerase to progress through this unsteady path, this mountainous path. These proteins will help this RNA polymerase to progress through mountainous path. Did you understand the difference in elongation? This RNA polymerase, eukaryotic RNA polymerase has more challenges for elongation. Now, did you understand? Did you understand the difference between elongation in prokaryote and eukaryote?
Yes, sir.
Okay, thank you. Now you can see this. So from this we will get the concept of heterochromatin and euchromatin. So there are two types of chromatin. You can see there are two types. Some regions, our all cells have same set of gene. Do you know this? All cells have same set of genes, but does all cells are similar? Are all cells similar? Tell me.
No.
No, but all same sets of genes. They are not similar. There are about 200 different types of cells. Why are they different? They are different because these cells, all sets of genes are present, but among this set, some sets of 4 will be, let's say sets of 4 will be active in one cell. Other sets of 4 will be active in other cell. Other sets of 4 will be active in other cells. Maybe some cells will have set of 5 genes will be active. The difference is because of though there is presence of all genes, but some genes are inactive in some cell, and the same type of gene will be active in other cell. And this gene activation and repression is done by this mechanism. This mechanism is called, some genes are switched off by this nucleotide, this nucleosome. Nucleosome will clump together, and when they clump together, they close this chromatin from reach of RNA polymerase. This chromatin from reach of RNA polymerase. Now, can you imagine RNA polymerase can transcribe this DNA here? That clumped DNA. Tell me, can RNA polymerase reach here?
No.
No. Can RNA polymerase reach here?
Yes.
Yes, because this is the basic difference between euchromatin and heterochromatin. So euchromatin can be transcribed. Transcription can happen for euchromatin. Transcription friendly chromatin. And this is transcription. This is not friendly for transcription. So euchromatin is friendly for transcription. Heterochromatin is not friendly for transcription. So this is the basic difference between heterochromatin and euchromatin. Heterochromatin is clump. Euchromatic nucleosome is a nucleosome of heterochromatin, and this is open nucleosome for euchromatin. This is open nucleosome of euchromatin, and this heterochromatin can be made into euchromatin. There are mechanisms in our body that can change heterochromatin into euchromatin. Okay, and there are mechanism. That mechanism will actually add something into this nucleosome. And then because of that, this will loosen. So I'm not going to talk about this mechanism. Just remember that there are mechanisms in our body that will cause this switched off gene to make it switch on. So switch off gene can be made switch on, and this switched on gene is euchromatin, switched off gene is heterochromatin. That is the basic difference between heterochromatin and eukaryotic. This is important concept. That is why I have to mention here. Now, so transcription happens in which chromatin? Heterochromatin and euchromatin? Yes.
Euchromatin.
Euchromatin. Transcription happen in euchromatin, not in heterochromatin. And that is sufficient for your information. If you remember these two structure, you don't have to go into detail. If you, for the time being, if you remember these two structure, then later you will understand the process of that conversion of heterochromatin into euchromatin. It's not that difficult. Now let's talk about this is elongation phase. So elongation transcription happens for only euchromatin region, and this is also through chromatin remotely by this, and then some other factor will help it. Now termination will be done by these two proteins. This is called CSTF and CPSF. This is called cleavage stimulating factor, and then cleavage and polyadenylation factor. You know, poly A tail is there in eukaryotic mRNA. Now this protein will be recruited to some region. There are region. This region is called cleavage signal, and that will be transcribed. After it's being transcribed, this will bind to this protein, two protein, and this two protein will cause cleavage and addition of poly A tail. So this is the termination in eukaryote, a bit different termination. So did you understand the difference between prokaryote and eukaryote also?